IGCSE Biology Paper 6, which is Alternative Practical Guide. The essence of this video is to explain all the major content that will definitely come out in an alternative practical exams. Since the exam is tomorrow, so this it's more like a day to exam revision guide that I love to go through with my student, and I believe this will actually help a great deal. Now, in this guide here is what we're going to look at. The major key points. We're going to look at how drawing of a specimen, the guide on what you need to do to get the complete mark. We're going to look at calculating magnification. We'll look at how an experiment, designing an experiment that will definitely come out as a 6-mark question. So we're going to look at how we identify major errors and their improvement, and we're going to see how graphs, some of the tricks involved in plotting graphs, like how you should be able to know what will be on your x-axis and your y-axis using the table in order for it to get at least the mark, the complete mark attached to it. Now, the major reason is this paper is a 40-mark question. So these are the major part, because drawing a specimen will come out as a 4-mark question, calculating magnification will be 3, and designing an experiment will be 4, identifying errors and improvements usually come out as 2-mark question. Then graph also come out as sometimes as a 5-mark question, including the conclusion. So if you add this together, this is like 10, 13, 15. This is already like 20 over 40. So you see, it's really easy. So these are the major aspects that if you know what you're doing here, you'll definitely be able to do the other 20 marks, because they are quite straightforward and the answers are what you can pick out from the question. Now, drawing a specimen in biology. Now, when you draw a specimen, the first thing you should go to the exam hall tomorrow with is you should make sure you go to the exam hall with a very sharp pencil. Your outline should be very clear, and the drawing should be as large as possible. So it should at least fill up the space given to you. You should fill up the space provided. Should fill up the space provided in the exam question. And now it has a definite outline. You should have a definite outline with no shading. There should be no shading, no matter what, because that already will make you lose 1 mark. And no arrows should be used in labeling. Let me give you an example. Possibly I draw as an animal cell and I want to label the nucleus. Then I do something like an arrow, whether in any direction, and I write a nucleus there. So this will definitely make you lose mark. So if you want to label, you should just point to the direction and draw a straight line and label your diagram. That is very important. Now, another major clue in drawing a specimen is that you should always try to be detailed enough. So you should check the diagram. If it's an internal structure, you should be able to count some of the major features that are there and include it in your diagram. You usually get 1 mark for that. Now, I have an example here of examiner's report or sample of student paper. Now, this particular student was able to score the complete 4 mark in this drawing. Now, because he was able to go through all the criteria that is needed. 1, the diagram is large. It covers more than half of the spaces given. Now, you can see that there are single lines with no shading included. There are no shadings here. Now, if you also look at the details I was trying to talk about, if you count the number of segments here, we have like 1, we have 2, we have 3, 4, 5, 6, 7, 8, like 9, 10, 11, 12, 13, and possibly 14. Then let's count his and see. He has 1, he has 2, he has 3, he has 4, he has 5, 6, 7, 8, 9, 10, 11, 12, and 13, which is similar to that in the diagram. So that is the detail I'm trying to talk about. If you see here too, he has like 1, 2, 3, 4 section. Here too is 1, 2, 3, 4 sections. So you should take note of this when drawing your diagram. That will actually help you to achieve the full marks that is needed in this question. Now, here is another example of a candidate response booklet, and this candidate is also able to get the complete 4 marks. Now, first, the diagram is very large as required in the syllabus, so it's very large, covers almost all the space provided. Now, if you look at the detail, there are also single line with no shading, single line, no shading. He also have sorry, if you look at the veins on this diagram also, there are like 1, 2, 3, 4, 5, 6, 7, 8. So he is so detailed, he was able to draw them completely. 3, 4, 5, 6, 7, 8. So when drawing this diagram in the exams you have tomorrow, you need to take note of these kind of details and include them in your drawing. Single line, large, for it to just be large alone is 1 mark. Single line is 1 mark. Detailed is 1 mark. No shading, you get another mark. So you definitely get your 4 marks. So this is just samples that I need you to take note of. Now, in size of a specimen, remember, your alternative to practical, they do not have a core or extended alternative to practical paper. So it is a single paper for both those that are writing core and extended exam. So everybody will take this alternative to practical component. So what that means is there will be no question tomorrow that will come and require you to measure the size of a specimen that will not be in millimeters. So what I normally tell my students is, always in the exam, every measurement you're going to do, try to always make them in millimeters. And if you don't have a millimeters ruler as you're going to the exam tomorrow, try to get a ruler that measures in millimeters. The major reason is, sometimes you know that to convert from centimeters to millimeters, you just multiply by 10. So if you have 1.5, you want to convert to millimeters, you multiply by 10, which will give you 15. However, 15 millimeters. However, sometimes you might know this conversion, and when you get to the exam hall, because of exam pressure, because of stress and the rest, you might just measure 1.5 in centimeters and you forget that you need to convert it to millimeters. So always try to remember that every measurement you're going to do in this exam tomorrow, your answers should be in millimeters. Every measurement. Now, next, we have a sample of how you answer this specimen type of question. Now, also remember that in alternative practical, the formula will be given to you. The formula will be given to you. So all you need to do is to substitute into the formula to get your required answers. So, but however, if you remember aim, you aim at something. AIM is to aim. So that's actual side, image side, and magnification. That's aim. I think some of my students used to call it the I am. So I am also, if you could remember this, then you should know that I come with am. Then you can substitute any of the formula you need to. But however, for this paper tomorrow, the formula will be given to you. So I have a sample here that said the magnification of the wood louse in figure 2.2 is times 9. So this is actually the figure here. I think I included it. This figure is 9. So the magnification is 9. Now it is required that we also try to calculate the, let's see, the question that is required. Sorry. They said measure the length of PQ. So all you need to do is you go to your diagram, you use your ruler and measure this length on the diagram given to you. So line PQ with your ruler and make sure you convert to millimeters. Like in this particular question, they often put the units for you at the end here, so you know it should be in millimeters. So in case you forget and you just write 4.7, you are going to lose any mark that is attached to this question here. So you should always remember that if you measure in centimeters, you multiply by 10, it will give you 47. But if you have a ruler that measures in millimeters, it makes things easy for you. Now they say calculate the actual width of the woodlouse using the formula and your measurement. So magnification, the formula is now given. See what I told you, formula is given. So magnification is equal to length of line PQ, which we measure to be 47, divided by the actual width, which is what we are looking for, is equal to magnification, and magnification is given as 9. Now, in some question, they won't give you magnification in the diagram. They won't give you it won't be written here as times 9. So all you need to do in those type of questions is to look at the diagram. You will always see the magnification written there beneath the diagram, and you see the multiplication sign in front of it. So all we need to do now will be 9 is equal to 47 over x. So your x, this will equal to 9x is equal to what? 47. So your x will be equal to 47 divided by 9. Then I think you get 5.22, because here your answer should be in 3 significant figures. So that will obviously give you a mark. So now designing an experiment. This is a 6-mark question. So you need to pay attention for all the details. I try to make it as simple as possible. Now, when designing an experiment in this alternative practical, some of the basic things you need to know is what is being listed here. First thing you need to do is to identify your independent variable in the experiment that you want to design. The first thing, you identify your independent variable. Now, what is the independent variable? The independent variable is the variable that is changed in the experiment. So it is what is changed in the experiment, what you change. So now examples, these are examples of how you change your independent variables. Like you prepare 4 identical test tubes with 5 different temperature. In this case, temperature is the independent variable. That is what you want to change. Now, the temperature in 10 degrees Celsius, you have 20 degrees Celsius, you have 30 degrees Celsius, 40 degrees Celsius, 50 degrees Celsius. So you are looking at 5 different temperatures. Sorry, this should be 5 identical test tube, because each of the temperature we have is own test tube. Now, use 5 different pH values. Then in this case, you find out that the different pH, that is what you want to change. Then you also look at this other example is you use 5 different plant species. So in that case, it is the plant species that you want to change in the experiment. Then the 4th one I have here is when you look at concentration of anything. So here it is estimated that you should use at least 5 different concentration of enzymes. So in that case, you put them in percentages. This will also go, or you look at different distance from light source in case you're looking at photosynthesis, and in that case light source is your independent variable. So these are some examples of ways in which you alter the independent variable that you identify in the research question, the question they want you to describe how you will plan an experiment or an investigative test for it. Now also, when doing that, the first thing is to identify your independent variable and tell the examiner how you want to manipulate it. Then after that, the second thing you should do is to list all variables you need to keep constant. You list it. Now note, the word amount, sorry, this should be WORD. The word amount is not accepted. So instead of using amount, you should use words like volume. You should use words like mass. Do not use amount. We are not looking at money here. We are looking at something we could measure in the lab. Now, examples of things that and ways you should keep things constant is that you use same temperature using a thermostatically controlled water bath. It helps to keep the temperature constant. You use same volume of water. You see, I didn't say same amount of water. So take note of these key terms when writing these exams tomorrow. Now, you should also use same pH, same surface area, same concentration of enzymes, same concentration of salt, sucrose solution, alcohol, iodine solution, Benedict reagent. All these are things you should keep constant, but all this is in line with the type of experiment that you are planning. It's not that you go to the exam hall tomorrow and begin to write all this. No, it is every experiment has its own stuff that you're going to keep constant, what you're going to manipulate, what you're going to measure. We're going to look at a sample shortly. So we say same mass of food test. Also very important. You check the mass. So it's the same. It's what you want to keep constant. So if it is plant, sometimes you can say same species of plant, same age of plant, same number of leaves, same light intensity, same water volume. So all these are things that you want to remain constant throughout the course of the experiment. Same gender, age, body mass, etc. Then dependent variable. Now that's what is kept constant, the constant variable, what was controlled. Now independent variable is what you measure, what is measured in the experiment. What is measured in the experiment is the dependent variable. Now, you should say how you will measure experiment and always and include the apparatus you use to measure. Include the apparatus you use to measure. Now, example is this. You take time taken for color to change. So the time taken is what you're measuring, and in that case, you use a stopwatch. Now, change in color, you use what is called a colorimeter or colorimeter. You use that to check the change in color. Now, volume of gas, when you want to measure volume of gas, you use a gas syringe. When you want to measure time, like I said here, you use a stopwatch. When you want to measure the temperature, you use a thermometer. When you want to measure the mass of anything, whether it's mass of potato, mass of anything in this experiment, always know that you use a balance scale. Now, there are other things you should measure when designing an experiment. You measure how long the experiment will last. Very important. It's 1 mark. If you want to keep it for 15 minutes, if you want to keep it for 3 seconds, if you want to keep it for 30 minutes, if you want to keep it for 3 days. So you should state the amount or mention the amount, or sorry, you should mention how long the experiment will last. Remember, tomorrow do not use anything amount, please. Now, other important stuff that should be included in your 6 mark question is the reliability of your experiment. And in that case, you should always try to say repeat experiment to get more reliable result, or you said repeat the experiment to get an average, repeat the experiment to exclude anomalies and minimize error. Then at the end of every investigation, we want to know your safety precautions. So in this case, it's things like wear lab coats, wear goggles, wear gloves, use tongs, and tie your hair back. So you have special clothing for exercise or something like a training shoe. Now here is a candidate, a sample response to a 6 mark question. They say a student stated that catalase activity is the same in all species of plant. So now, if we're looking at this, you find out that what you need to change to test this experiment, because they want you to plan an investigation, is the species of plant. So you need to get at least 3 different species of plant or 4 different species of plant. So in that case, you find out that this species of plant is your independent variable. So now look at how it was changed. Choose 4 different plant species. So he has mentioned his independent variable, and remove 2 leaves from each plant species. Now, 2 leaves from each plant. So each plant you remove 2 leaves. That's also a constant now, or control variable. Grind the leaf up in a mortar and a pestle. Use spatula to add some of the ground up leaves to 10 cm cube of hydrogen peroxide solution. So this 10 cm cube of hydrogen peroxide solution is also a constant or control variable in the test tube. Now leave it for 5 minutes, so the time taken for the experiment, and then measure the height of the foam with a ruler. So he's measuring the height of the foam with a ruler. That's his dependent variable. Now he said record the height of the foam in millimeters. He also explained how you should record it. Use exactly the same procedure for all 4 test tubes. So for all 4 different plants. So here you have 4 different plants. So the same procedures is used for the 4 different plants. Then after that, he said 4 different plant species. Then repeat the whole experiment and take average height. So repeat and take average height is his reliability. Now, for each species can be calculated. Hydrogen peroxide solution is dangerous. So gloves and goggles should be worn. So that is the safety. So you see, all the criteria that you need in an experiment is here. So this particular candidate will get a complete 6 mark for designing his experiment like this. So first, you choose 4 independent variables. You state the things that you want to be constant. You state how you want to measure and how you will record the measurement. That is it. And the instrument is here. So dependent variable is what you measure. So he is able to look at all the criteria that is needed in planning an experiment in an exam. Now, while writing this exam tomorrow, in case you will be confused in trying to structure your answers like this, I also tell my students you should you can actually start by writing independent variable as a subheading. Then you write control or constant variable as the second subheading, and you write what you want to control. Then you now come and write the procedure. Then it will make it easy for you to now write the necessary explanation, because you have your independent variable and your control. So you just be able to write it in a form where everybody will understand. So it's quite that it makes it really easy. Then the next thing after procedure, you can come and write your dependent variable. So you tell us how you measure it and the instrument you use. Then you now come and write reliability, which is always repeat and take average. Then you write your safety. So you see, this is 1 mark, 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 this is 1 mark. You definitely get your 6 marks. So next is identification of errors. Identification of error in an experiment. So it comes out like wildfire. So these are samples I just gave. So when you read through the steps in this exam tomorrow, you should be able to identify errors in each steps. Now, what this material will help you to do is to give you examples of some of these errors and how you can possibly improve on it. Because some questions we just ask you to identify errors. Some questions we always ask you to identify errors and how you should improve on it for a better result. Now, one of the major errors in most IGCSE paper that come out a lot is experiment will always be done once. So if experiment was done once, the improvement is that you should repeat the experiment and take average. Now, when the water temperature changes, so at the beginning, possibly they did not use a thermostatically controlled water bath, so that should be the improvement. If they don't mention it there, even if they mention water bath, as long as it's not thermostatically controlled, you expect that by the end of the experiment, the temperature would have decreased. Now, next, you look at some bubbles are larger than others. Now, there can be miscount of bubbles because we are humans. Now, the best way you collect volume of gas, the improvement is that you should use a gas syringe to measure volume of gas. Now, water can easily be lost through evaporation if it's just left in a beaker or in a test tube without it being covered. So in order to prevent water loss, it should be covered to prevent evaporation. So take note from the experiment. Did they cover the test tube with? If they don't cover it, then there is high possibility of evaporation. So one way to control that is for you to cover to prevent evaporation. Now, here, reuse of syringe. Take note, these are very common errors that we've seen over the years in IGCSE alternative practical. Now, reuse of syringe, reuse of glass rod. Sorry, there's no E here. Reuse of glass rod. This will lead to contamination. If you use them over and over again for the same test tube, it can lead to contamination, therefore affecting the result. So what the improvement is that you should use new syringe or glass rod each time you're carrying out an experiment. Now, color change can be subjective, because if we are observing color change as humans, some people can be visually impaired, so they will not be able to give the exact color change. So the best thing you should do when looking at, if the experiment is looking at colors, you should use a colorimeter to always check if the color, other than using human subjective ability, which is subjective to error. Now, we also look at measuring the height of water is inaccurate. Instead of measuring the height using a ruler, it is better than measure the volume. So take note of these things in the exam. Now, also dropping or pouring solution can make the volume to vary. So the best way you do it is you use a graduated pipette or a measuring cylinder to make sure the volume of everything you're doing is equal. Now, shaking of solution, if you shake it, it can actually lead to spillage. So you check within those steps as they are describing it in the experiment, uh, what did they mention? So sometimes they use these terms and they will ask you to identify the errors. So shaking of solution can cause spillage, and the best way, instead of shaking it, you should use a glass rod to stir the solution. Now, gas can also escape, so usually you seal it tightly to prevent that. Now, these are some of the errors and improvement. Now, graphs in IGCSE. There are skills you need to know. Now, the first thing is you will definitely be given a table. And once you're given a table and you're told to plot a graph, some basic thing you should know is you can be asked to plot a line graph tomorrow. You can be asked to plot a bar chart or possibly a histogram. Outside these three, none. And also you should learn about a line of best fit. Now, where most student interchanges, line graph is quite straightforward. They don't make mistake of that with that. But where most of them make mistake is between bar and histogram. Now, a bar chart usually have space, equal space in between each bars. Then a histogram is when you have no space and all of them are quite joined together in this format. So take note of that. Now, if you are given a table like this, where most students have issues is that you know that if you're given a graph, this is your x-axis and this is your y-axis. They now begin to ask themselves, which of these value will be on my x? What I normally tell my students is, if you are saying your ABCD, A, B, C, D, E, F, G, H, I, J, K, L, M, O, P, Q, R, X, T, U, V, W, X, Y. So X always come before Y. So if you are given the table, the first column is usually your X. Then the next one you want to plot against is usually your Y. X always come before Y. And with that being said, your X is always your independent variable and your Y is always your dependent variable. Take note of that. So all you need to do first to get your first mark is to copy everything. Do not leave any single thing. Everything that is here, you copy them and write it under your x-axis. That is the label. That is 1 mark. Everything that is on your y, you copy it, everything the way it is, and write it under your y-axis. Then you now come to look at your scale to now do your plotting. Now, for this particular data, the plot is here. So as you can see, the first thing the student was able to do, he wrote his number of bubbles, which is what is here. But he did not copy everything that is here. He only just wrote number of bubbles. And because of that, he lost 1 mark. While on his x-axis, he was able to copy everything that was here and he included it here. But the 2 is needed for you to get your 1 mark. Now, however, the scale was properly used and also for both the x and y, and everything was plotted properly. Now, but if you check this, the examiner, this is more like the examiner's comment. He said the candidates include all appropriate scales on their graph, plot all points correctly, and draw a reasonable line of best fit. Now, when drawing this line, make sure there are no cut intervals. You don't draw straight and join this, then pause and join, and then pause and join. No, you should make sure they are all joined together. Very important. And draw a reasonable line of best fit. It would be better to label the y-axis as number of bubbles in 5 minutes. So you see, rather than just number of bubbles. So mark is 3 out of 4 just because of this mistake. So once you get to that exam hall, make sure everything that is on the heading that is on x-axis, you write it on x. Everything that is on y-axis, you write it on your y. Now, lastly, take note of this, please. Food test. It definitely will come out. You might see 1 or 2 or 3 of it as much as possible. It comes out in between questions. They have a way of bringing in food test into the question. Now, if you look at this food test, they said, how do you test for starch? You add few drops of iodine solution to the food sample. Positive test, it will give you blue, black, and negative test will remain brown. Testing for reducing sugar like glucose, sucrose, amino acid. How you test? Sorry, not amino acid. Sorry, amino acid has to do with protein. That's an error. Amino acid is protein. Glucose, sucrose, maltose, they are all reducing sugar. So in testing for reducing sugar, you add Benedict solution to the food sample. Then you heat for 2 to 3 minutes to at least 80 degrees Celsius in a water bath. Now, once you do that, you now observe the color change. If it changes color from blue, because Benedict solution is blue, so if it changes from blue to green or red, depending on the concentration of uh, reducing sugar, that's positive test. Then if you're testing for protein, you add few drops of burette reagent. Now, positive result, it will give you a mauve or purple color, while negative result, it will remain blue because burette is blue. Then test for fat. In testing of fat is the emulsion test, where you add ethanol to food sample, then you pour into a test tube with an equal volume of distilled water. That's, you pour the ethanol and food sample into a test tube with equal volume of distilled water. Now, positive result is that it will form a milky white precipitate. Then lastly is vitamin C. How you test for vitamin C, you add what is referred to as the DCPIP blue dye solution to a food sample. Now, positive test is that the blue dye DCPIP solution will become colorless. So please take note of everything I've explained. Make note, listen to them carefully. There is no how with what I've just said that you will enter into that exam hall tomorrow and score less than 30. It is impossible. So have a nice day and good luck in your exams.